I'm Clara, and I'm working on the development of new in vitro systems to be able to test drugs on them. We are going to try to build human organs, and we are going to build it on small devices that are called chips. The idea of these systems is actually to do more physiologically relevant models where we can test drugs, get answers before, and by this way speeding up the process of drug development, but also reducing the tests that we need to do on animals. I am a biomedical engineer by training. I came to France and I did a master on biomechanics, where I discovered a bit the world of organs on chips. And then I did my PhD thesis also working on organs on chip, trying to develop the blood vessels from the brain on chip. During my postdoc, I have two main projects, both working on an organ on chip. So here at Servier, we are working on the liver on a chip, and we are going to use two different platforms. In one of these platforms, our aim is to characterize the liver on chip, see if it's functional, for how much time we can have it on culture, and also we want to see if we can help all the other departments. For example, we want to see if we can extract the genetic information to see if a drug is going to make a change on the livers. Basically, we're going to try to see all the information we can extract from these systems to be able to introduce this liver on a chip onto the drug development cycle. And then in the other hand, we have another project using another system where we want to homogenize and to make sure that we can use this system for drug development. We're working with other pharmaceutical companies to make all the same protocol and to see if they are reproducible and if we are giving a plus to a standard in vitro systems. Basically, the experiment starts when we ask the question of what we want to do, design the experiment, and then we can move to the lab when everything is ready. We do our experiment, and then we move to get all the possible readouts, do the analysis, and then in theory we will be answering our question. One of the interesting questions of working with these devices is that actually they are not a standard. So sometimes we need to think on how are we going to be able to read our results using the traditional system. So we need to adapt the system a bit and think of different manners on how can we put our new technology on what it already exists. My department, which is the DMPK, actually is involved in all the phases of drug development from early research to clinical phase, because basically what happens in my departments, we are following the drug since it enters into the body until it's secreted. There are many molecules at the start, but not many that finish. So I think it's quite interesting because we are really close to the patient and we feel that we have a real contact and we are doing something for them. It's easy to work here and comfortable. There's another important fact that we are all the teams on research mixed, so it's quite easy to go and talk with another department that if it would be not here at SECLE, you would not be able to do the interaction so easily. And then also being here at SECLE, we are surrounded by many of great universities that we have here in the Paris region, and there are many academic labs doing research that are also of interest for us. So we can do many collaborations, we can go to their labs and see how can we help each other. I always wanted to do some engineering part mixed with medical sciences, and then I mix them. I try to start a mix of them, and then by doing some internships I just discovered would I like it or not, and the day I discovered organs on chip and the possibilities that we can do with them, I really enjoyed. And then I came to pharma, and then I started discovering the world of pharma, which is quite interesting, and you can see that you can have also a real impact on people's life.